everyone, and welcome to today's Clinical Labs webinar series. My name is Mary Beth Didon, and I'll be moderating this discussion, Optimizing Genetic Health Risk Testing, the Power of Pre-Analytical Systems. We like our webinars to be very interactive, so we encourage you to submit your questions to us at any point during this webinar. Our speakers will address your questions during the Q&A session following their presentation. To ask a question or leave a comment, simply type your query into the Q&A box on the right side of your screen. We will try to address as many questions as possible during our time together, but if we run out of time, I will forward any unanswered questions to our speakers and they can respond to you directly if possible. Please see the Handouts tab on the right side of your screen for supporting information for this event. I'd like to remind you that this webinar recording will be available on demand shortly following this presentation, so please watch for an email from today's Clinical Lab on how to access this free video once it's available. Our presenters for this webinar are Tara Crawford-Parks, PhD, Director R&D with DNA Genotek, Jonathan Chan, BSc, Program Manager, Regulatory Strategy with DNA Genotek, and Shelley Gunn, MD, PhD, Founder and Medical Director of Targeted Genomics. Thank you to our sponsor, DNA Genotek. Their support allows today's Clinical Lab to offer these webinars free of charge to our audience. Everyone, thanks for joining us. Tara, please get us started.
Good afternoon, everyone. My name is Tara Crawford-Parks, and I'm a director in the R&D department at DNA Genotek. Today, we have a really great webinar planned for you to provide insights into the value that our DNA Genotek products can bring to the genetic testing market. Our portfolio of sample collection devices enables the easy self-collection of samples either in the clinic or at home, targeting minimally invasive and comfortable user experiences. We have devices available for collecting a range of sample types for human genomics, epigenomics, and microbiomic use. Many of our devices have marketing authorizations for IVD use from regulatory agencies in Europe, the UK, and the US FDA. Today, I'll focus on two of our sample collection devices. Our saliva collection devices are designed with user experience in mind, and superior user comprehension and compliance have been demonstrated through robust usability studies. So we have the OraCollect DX device, which is shown on the left, which uses a soft sponge tip to collect saliva and provides a more comfortable collection for the user. The Oragene DX device, which is shown on the right-hand side of the slide, is a direct spit device that collects 1 to 2 milliliters of liquid saliva. It's designed with a wide funnel and a fill-to line to ensure that the user can easily collect the proper amount of saliva. Both devices contain a DNA stabilization chemistry that prevents DNA degradation and bacterial growth, enabling ambient temperature storage of the sample from a user's home to the laboratory. Saliva as a sample type offers some unique characteristics to consider when deciding which sample type to include in your assay. It provides a good source of nucleic acids, both genomic DNA and RNA, and including small non-coding RNA. DNA extracted from saliva has been used in a variety of applications, including cancer, epigenetics, and diagnostic applications, including the rare disease testing field. The DNA in saliva comes primarily from the white blood cells, with a smaller proportion, which is about 15%, from the buccal epithelial cells. The figure in the center of the slide shows the various cell types that are present in the saliva, and the bacterial DNA content of saliva collected using our Oragene or OraCollect devices is about 5% on average. This is a much lower amount than oral collection devices such as a buccal swab or cytal brushes. I will now pass it over to Shelley from Targeted Genomics to share some insights they have from developing a genetic health risk test using saliva as a sample type, and the benefits of leveraging our OraCollect DX device and its FDA general clearance.
Thank you, Tara. My portion of this presentation will describe how integration of OraCollect swabs into our testing system improved our device and shortened our timeline as we navigated our first product through the FDA clearance process. And this is an overview of what I will talk about, starting with the value of genetic health risk tests to help guide personalized healthcare. And throughout the presentation, I will use the acronym GHR test instead of trying to say genetic health risk test through this whole presentation. We'll be talking about the advantages of saliva as a sample type to facilitate at-home sample collection, and why we chose to seek FDA clearance for our GHR test, which is called Gluten ID. So Gluten ID is a GHR test for celiac disease, the most common inherited autoimmune disease in the United States. And in these statistics from the Celiac Disease Foundation, there are an estimated 3 million Americans living with celiac disease, and yet 2 million of that 3 million don't know they have it. Also keep in mind, the estimated 3 million people with celiac disease all have families. And celiac disease runs in families. And this graphic shows why celiac disease has been called a silent epidemic. The real number of celiacs in the United States might be better estimated by multiplying 3 million times the number of individuals in each one's family. But once an individual has been diagnosed with celiac disease, the whole family is at risk until proven otherwise. However, not everyone is truly at risk. Only individuals who inherit the HLA DQ2 and/or DQ8 genes associated with celiac disease can develop the disease. No risk genes equals no celiac disease. So in the family you see illustrated here, the two blue figures have been diagnosed with celiac disease, putting everyone else at risk. But how would you know which of these families members are actually at risk? So it was to answer this question and address the hidden epidemic of celiac disease, as it is called, that we developed the Gluten ID Celiac GHR test. We also developed a physician-ordered test based on the same assay called Celiac DX, and this is a physician-ordered, pathologist-reviewed test. But both of these tests detect all possible combinations of the celiac risk genes from an at-home collected saliva sample. The next generation sequencing assay that we designed to test the DQ2 and DQ8 genes was developed at ResearchDx in Irvine, California, where I'm the chief medical officer. And the test is now run in our CAP-CLIA certified clinical laboratory called Pacific DX. And if you want to read more about celiac disease, I highly recommend the book that's illustrated on this slide, which is by Peter Green, who is the director of the Celiac Disease Center at Columbia University. Celiac disease only occurs in individuals who have inherited the DQ2 and/or DQ8 celiac risk genes. And as shown in this pyramid, gluten is the trigger, but celiac disease won't develop without the genes. So that's why genetic testing is so important for celiac disease. Individuals who don't have the celiac risk genes can rule out celiac disease with close to 100% certainty for the rest of their lives. So now about our test. Here's a direct-to-consumer workflow, which, as you would expect, begins and ends at home. Gluten ID kits are ordered online and sent directly to users at their homes or office. And the test kit basically consists of an OCD 100 saliva collection device, instructions, and a USPS mailing label for sending the sample back to our laboratory in Irvine, California, which is illustrated in the picture at the top. Following DNA extraction and library preparation, samples are batched and sequenced on the MySeq DX, which is also a Class 2 medical device. Gluten ID is designed to be a high-throughput sequencing assay. We can run 90-plus samples at a time with a 2-day turnaround time. However, we batch our samples in order to fill our 96-well plates, because we do have some controls on the plate as well. So the actual turnaround time for users is longer than 2 days. In fact, it can be several weeks depending on how many samples we have in the lab. The raw sequencing data is then converted to tech-only data and then converted again through our proprietary Gluten ID software to a result-specific report for each user, and it is then sent via secure file sharing directly back to the consumer. So as you can see, this process starts at home and it ends at home with delivery of the report. And this is an example of a risk gradient from one of our gluten ID report templates. There are 15 possible DQ2, DQ8 gene combinations and gluten ID tests for all of them. The results are reported within a risk gradient, which is shown here. And this is illustrating a high-risk result. So the higher the risk, the higher the genetic predisposition for celiac disease, and the more likely an individual is to develop celiac disease during their lifetime if they're eating gluten. And these are what we call our gluten ID risk meters from the report. The one on the left is showing a result where no variants were detected, not inherited from either mom or dad. So this can virtually rule out celiac disease. The result in the middle is a low risk result. And then on the right, this report template shows the risk meter when a celiac risk gene has been inherited from both parents. So we included in our 510K submission to the FDA a case report to illustrate for our review committee the clinical utility of the gluten ID test as an at-home test for a family. And the quick summary of this case report is it's a 27-year-old previously healthy male with severe acute gastroenteritis, which is indicative or suggestive of actually something in a medical emergency called celiac crisis. He was admitted to UC Irvine ER with symptoms that needed to be immediately addressed. And he had no family history of celiac disease, but the symptoms told the story. And after TTG antibody testing and small bowel biopsy, which are the diagnostic tests for celiac disease, he was confirmed to have celiac disease. The patient, who was actually a technician in our laboratory in Irvine, was quite concerned about the rest of his family. And so we provided gluten ID kits for the entire family. And the genetic results from his family, by the way, samples were all collected at home and then sent back to the laboratory, just as any at-home testing would be, showed that 4 out of 5 members were indeed at increased risk for celiac disease. So the patient is now healthy, eating gluten-free, as is the rest of his family, and a gluten-free diet is the only, or currently the only, effective cure for celiac disease. Of note, a gluten-free diet can also be preventative for those at risk, because with the DQ2 and DQ8 genes, gluten could very well trigger celiac disease in those individuals. So based on this case report, we provided the FDA with rationale in our 510K submission that as a direct-to-consumer test, gluten ID has the advantage of consumer choice. As you saw, this family chose to test everyone. They collected their samples at home, and then we provided them with comprehensible reports that were designed to share with their healthcare providers to decide next steps as to how to manage their increased celiac risk. I'd also like to call to your attention on this slide the indications for use in our first version of the 510K, this is our eStar that we submitted, showing that the collection does not specify the collection device, but said that we were going to use genomic DNA isolated from human buccal cheek cells, and we will get back to what happened with that. Our further rationale for submitting a 510K instead of keeping our test as an LDT was the uncertainty surrounding the FDA's final rule on LDTs. This was all brewing while we were transitioning our test from an LDT into a Class 2 medical device, and it really served to help us continue on that path, not knowing how that was going to turn out. I believe it's resolved for now. But also another rationale was the ability to do diagnostic testing on an individual basis as well as for the family. So that's not usually done with large laboratories and in the prescription world that we live in. If a decision is made that an individual in a family has high risk and the rest of the family needs to be tested, this allows the family to do that quickly and in the privacy of their home. We also liked the fact that going through the FDA would give us the opportunity to do user studies and really transform our reports from just a basic reporting of results into a comprehensible report that could be understood by the majority of people in the United States. So here is our submission timeline. We submitted our original 510K in 2022 in July after 2 years of optimizing the LDT and then doing our analytical performance studies over those 2 years. And in November, specifically November 2nd, 2022, we received our additional information letter from the FDA. And instead of having clearance, we needed to provide a lot of additional information to the FDA. And the major topic, the major deficiency was that as they said, it appears that the study you provided in support of your clearance was collected with a collection device that does not conform to the specifications under 21 CFR 866-5950, which those of you who have submitted a GHR test to the FDA can sympathize. So submission hurdles in an FDA clearance journey are significant, especially with a genetic health risk device, which is further regulated by special controls. One of those special controls is that the GHR device must use a Class 2 sample collection device. Our original buccal cell collection device was Class 1. So our FDA review committee presented us with 2 options. We could submit a separate 510K for the current collection device, or we could perform new analytical studies using an FDA-cleared collection device, and one that was cleared for over-the-counter use in molecular genotyping testing, by the way. Option 1 was not an option for us because we didn't have the resources, so we chose option 2. A new collection device. But what collection device could we use? So OraSure to the rescue. And they actually announced very soon, actually the day after we received our AI letter, on November 3rd, 2022, that they had received FDA general clearance for their OraCollect device. And so we proposed to the FDA that we would change the gluten ID collection device to OraCollect, and they found that proposal to be very acceptable. So the only downside for us was we had to redo all of our analytical performance studies, but the good news was with our new collection device from OraSure, all of the results were 100% concordant with the new device. So this is our finalized indications for use that became official on January 7th of this year when our device was cleared. And they are very specific for the OCD 100 collection device, must be used with our gluten ID kit. So to summarize, the advantages, which are many, of integrating OraCollect FDA-cleared device into a test system is obviously the time and money saved by not having to submit a separate 510K. And then the packaging and presentation of the device is really just perfect for a kit such as ours. The instructions are immediately available on the package. We repeat those in another instructions booklet in our kit, but we've had only good things to hear from our users that it's an easy collection procedure for them. And then our laboratory is happy because the collection device fits seamlessly into our laboratory workflow and DNA extraction procedures. And then the satisfaction that users have with all at-home devices that are bloodless, pain-free. Saliva is really the ideal sample at-home collection method. So with that, I would like to turn it back over to Tara and let her tell you more about the saliva collection devices.
Thank you, Shelley. That was really interesting to hear about your experience that you've had in taking your test through the FDA, and exciting to learn more about the real-world challenges that your gluten ID test provides a solution for. As Shelley and I both mentioned, there are several advantages to incorporating an FDA-cleared sample collection device into a pre-analytical workflow of tests such as the gluten ID. Both OriGene-Dx and OraCollect-Dx have prescription and direct-to-consumer use within our FDA general clearance, which really positions them well to be used by commercial partners to be validated within their specific tests. There are several examples in the market today. Shelley just gave us a really great overview of how Targeted Genomics is using it, and there are other assay manufacturers that are using these products today in the market in their pre-analytical workflows, such as Helix and Grifols. Both the OriGene-Dx and OraCollect-Dx devices have been robustly validated for ease of use of sample collection. Specifically, our studies really focused on the product's use across naive user populations that were reflective of the general US population. These studies were designed to evaluate the comprehension of the instructions for use and compliance with returning the collected sample collection device using standard postal services. So really simulating that at-home self-collection environment. As an example of the type of data that we collected during these various usability studies, I'm going over the next few slides. I'll walk you through some of the key findings of these studies that we conducted for both OraCollect-Dx and the OriGene-Dx devices. To evaluate the use of OraCollect-Dx in a DTC environment, naive participants were recruited and provided a study kit which contained the OraCollect-Dx device. They were instructed to register the kit, collect a sample following the instructions for use, and then they were asked to complete a comprehension questionnaire on the instructions for use and any ancillary labeling, and to return the collected sample through the standard postal service to a processing lab where those samples would then be processed. To briefly take you through the collection steps here of our OraCollect-Dx device, you can see that on the right-hand side, you open up the packaging and you insert the sponge into the user inserts it into their mouth. They collect on either side of their lower gums to soak up the saliva. They then remove the swab from the tube and invert it, placing the swab into the DNA stabilization chemistry that is contained in the tube. And then you seal the tube by screwing on the cap. They were then instructed to place that collected sample into a biospecimen bag and then put it back into the mailer box and follow the shipping instructions to send it back to the lab. Once the samples were received at the lab, DNA was then extracted and evaluated for DNA yield and processed on downstream molecular diagnostic assays. The results from our studies demonstrated that 95% of participants complied with the shipping instructions and correctly answered the comprehension questions that was provided in that questionnaire. In addition, more than 99.5% of the samples met the DNA yield criteria with successful performance on downstream assay results. Similarly, the OriGene-Dx usability study, which I'll go through on the next slide, was designed very similarly to recruit naive user participants, which were then provided a study kit containing the device. The participants collected a sample following the instructions for use. They completed a very similar comprehension questionnaire related to those collection instructions, and then followed shipping instructions to send their sample back to a processing laboratory using the standard postal services. Similar to the results that I described on the last slide for OraCollect-Dx, this study for OriGene demonstrated compliance of participants with our shipping instruction, high comprehension of the IFU-related questions in that questionnaire, and the collected samples met the input criteria and performed successfully on the downstream PCR and NGS-based genotyping assays. Together, these studies really provide a robust data set to support the use of the products in an at-home and self-collected environment, which really provides a flexible option for where the samples can be collected and supporting a variety of needs of downstream assay manufacturers to really leverage the device as it fits your needs. In addition, there are other important aspects to consider, more so for commercially marketed tests, to ensure that there's a really streamlined end-to-end operational workflow, as well as brand recognition. This may be of particular interest, for example, for genetic testing companies whose target market may be a consumer who's interested in their genetic predisposition, for a particular disease, similar to what Shelley walked us through with their gluten ID test. In this regard, we also offer flexible customization services, which include design and branded materials, such as a custom instructions for use with a company logo and color palette, and custom branded packaging options, which can enable companies to really create a solution that serves the entirety of their workflow. In this customization, there's options, for example, shown on this side of the slide for Origin DX. You can see that there's various user touch points where branding can be placed. And in addition, the customization options really extend down to the saliva sample itself. We have options for customized barcode solutions, QR codes, secondary identifiers, accessory barcode labels, the list goes on. And with one purpose in mind being that we aim to provide a flexible solution to allow for donor privacy while linking individuals to their collection devices through allowing tracking through the full chain of custody, from a shipment to the donor's home, back to a processing lab, and back to the company for their final results. For companies requiring logistical support to enable their diagnostic tests, we also have single order fulfillment options and kitting services available, which is something that you can reach out directly to us for more information on. I will now pass it over to Jonathan to discuss some additional considerations from a regulatory strategy perspective and to dig more into the benefits of using an FDA general clearance device as you're thinking about incorporating a sample collection process into your assay.
Thank you, Tara. So for the next couple of slides, I'll be talking about how to integrate sample collection devices into your test system, including the benefits of using an FDA-cleared sample collection device and some important factors for you to consider when you're developing your regulatory strategy. Now on this slide, I want to highlight some of the benefits of integrating our FDA-cleared collection devices into your test system, especially if you're planning to go down the journey with FDA to obtain a clearance or an approval. So one benefit of using our devices is that they're cleared for both prescription use and over-the-counter use. So what this means is that our collection devices will be compatible with your test, whether it is prescribed by a physician or sold directly to consumer. Another benefit is that the performance of our devices have been demonstrated on multiple cleared assays and diagnostic tests. Some of these tests include Helix's Genetic Health Risk App Test, Grifols Alpha ID at-home genetic health risk service test, and of course, targeted Genomics gluten ID test. To this date, Origin DX and OraCollect DX are the only saliva collection devices cleared for general use and are incorporated in multiple diagnostic test clearances. These multiple clearances show just how well our devices work with a wide range of tests, so you can feel confident using our devices without worrying about compatibility or performance. A third benefit is that our devices are suitable for at-home self-collection. Our devices are non-invasive and easy to use, and as Tara explained previously, through our usability studies, we've demonstrated that patients can collect at home safely and effectively without supervision, and those studies have been reviewed and accepted by FDA. Therefore, integrating our devices with your tests will ensure proper sample collection and enhance the at-home user experience. Now, because our devices are cleared for prescription and over-the-counter use, and the performance of our devices have been demonstrated on multiple FDA-cleared assays, this means that no additional testing is required for the sample collection phase of your test system. As Shelley highlighted in the hurdles targeted genomics had to face in their FDA journey, if you plan on selling your tests directly to consumers, FDA expects that the saliva collection device that you integrate in your test system has already been cleared for over-the-counter use, or that you provide FDA with collection device usability study data that Tara highlighted previously before FDA can clear your test. Using a device like ours that has already been cleared for general use allows you not to worry about demonstrating performance of the collection device to FDA, and you can focus solely on providing the performance of your test. And one last thing I want to highlight is that because Origin DX and OraCollect DX are Class 2 devices, they're required to be GMP compliant. RUO saliva devices and some Class 1 saliva collection devices are exempt from GMP requirements. Although these devices may comply with these requirements, our devices are required to do so and are routinely audited to ensure and demonstrate GMP compliance. This means you can trust that our devices are manufactured in accordance with strict quality standards. Now, it's possible to integrate a sample collection device that hasn't been cleared for your use case. However, because you need to show performance of your test from beginning to end, including the performance of your collection device, this adds additional cost and time to you. It can be challenging when you have to rely on the collection device manufacturers to support you in providing the data. So using Origin, DX, and OraCollect eliminates all of those challenges and saving you time and money getting your test FDA cleared or approved. Now, if you decide that your goal is to obtain an FDA clearance or approval for your test, it's extremely important that you think about the regulatory requirements and develop your regulatory strategy as early as possible to ensure that you're successful in achieving that goal. Now, in this slide, I've included a few things that you should consider when you're developing that strategy. You'll need to consider your indications for use and the analytical and clinical application of your test. This will determine the classification of your device and the types of studies you'll need to run in order to demonstrate to FDA that your test is safe and effective. You'll also want to research and see if there's any existing tests or predicates out there. This will determine the type of submission you will submit to FDA and also help you understand the types of studies that FDA is expecting to see for your test. So identifying the appropriate type of FDA submission early in the development of your test is essential for accurately estimating the cost and effort required to achieve FDA clearance or approval. Now, you also want to consider your short-term and long-term goals, including your target market and the time to market. Now, in this webinar, we've focused mainly on the FDA, but if you have future plans to sell your tests outside of the US, you'll want to consider your target market as early as possible and see if you need to design your studies around country-specific regulatory requirements. One common mistake that we've encountered and experienced ourselves is neglecting to account for other countries in the regulatory strategy and also failing to design our studies to meet those unique country regulatory requirements. So this resulted in the need to conduct additional studies, leading to unnecessary delays and cost. Now, on the flip side, it's also important to understand your time to market because building a study that meets multiple geographical requirements may not be the best approach, especially if it delays your product launch in key markets. So that's why it's really important to clearly define your short-term and long-term goals and determine what is the most suitable approach for meeting those regulatory requirements while achieving your objectives. And lastly, you want to identify the resources and expertise that you have to reach your end goal. You may need to reach out to consultants or work with other companies that have the expertise to help you get your tests cleared and approved. Now, a benefit of partnering with a company like ours and integrating our devices into your test system is that it can definitely increase your chances of obtaining FDA clearance or approval successfully. We have over 15 years of experience in developing collection devices that have gone through FDA's rigorous clearance process and have collaborated with many test developers such as 23andMe, Helix, Grifols, and Targeted Genomics to support them in securing FDA clearance for their tests. And then also in terms of resources, one thing I would highly recommend is that you engage with FDA as soon as possible. You want to build a positive relationship with FDA and understand their expectations to ensure that you develop the right studies for a successful FDA clearance or approval. FDA's goal is to ensure that your tests in the market are safe and effective. And as long as you demonstrate your performance in accordance to their requirements or expectations, there's no reason for FDA to not clear or approve your test quickly. So don't be afraid to engage with FDA because they ultimately want you to be successful in bringing a new test that's beneficial to everyone. Now, if you have any questions about how to integrate our saliva sample collection devices with your test system or would like to request samples of our devices, you can contact us at info@dnagenotech.com. And if you have any questions about Target Genomics gluten ID test, you can contact them at customer support at targetedgenomics.com. And that concludes the main part of our presentation. Before we move to Q&A, we would like to thank you for your time and attention and hope this presentation has provided some valuable insights on the value of an FDA-cleared genetic health test and also some of the advantages of integrating an FDA-cleared saliva collection device into your test system.
All right, wonderful. Thank you very much to our speakers. We are about to move into our audience question and answer session. A reminder that you can submit your questions and comments by typing them directly into the Q&A box located on the right-hand side of your screen. Please also see the handouts tab on the right side of your screen for supporting information for this presentation. And don't forget to answer our special audience polls if you haven't already done so. So I'll invite our speakers to participate in our live audience Q&A at this point. So let's take the first question from our audience. Thank you all again for a great presentation. The first question from our audience here says, why was saliva chosen over other sample types for the gluten ID test? And how does it compare in terms of DNA quality, yield, or patient convenience?
Well, the main reason we chose it, I'll speak for gluten ID since we made that decision. Early on, we wanted saliva. We started with buccal cells, but as Tara pointed out, the collection device we ended up with is also including buccal cells within the saliva. We wanted it to be painless and simple. There are celiac disease tests out there for at-home use that are Lancet-based, but we wanted a simple, painless experience for our users, and also wanted to be able to use the test at home in all age groups, starting with infants. And so for that reason, we went with actually the OraCollect DX soft sponge device so that it makes collection simple for any age group. In terms of the laboratory, there is no difference in quantity or quality of DNA that we have found extracting from a saliva-based collection device versus a blood sample. So we've been really very pleased with that decision.
All right, wonderful. Thanks so much. We have a few other questions from our audience coming in. This one says, what specific validation studies were required when switching to the OraCollect DX device, and how did the results compare to the original device?
So the answer is all of the studies. We repeated everything, but it was a good exercise too, because it gave us a lot of confidence that what we had reported to the FDA with the previous collection device was accurately represented by the new collection device. So it was all the precision reproducibility studies and extraction study, analytical specificity in terms of interfering substances, and then lot-to-lot precision and detection limit were all repeated with excellent results.
All right, fantastic. Thank you. Here's a question that says, how does the gluten ID test ensure accuracy and reproducibility across different laboratories and sample batches?
Well, I'll answer that one too. So gluten ID is run at a single site laboratory. So Pacific DX in Irvine, California runs all of our testing. Accuracy was initially evaluated by the FDA with what they like to see in their analytical performance studies for next generation sequencing is a comparison to bidirectional Sanger sequencing. So we did over 200 samples to show 100% accuracy between the two methods. And then currently when we run the test, it's run, as I pointed out earlier, in batches of 90 samples plus controls. And it's those controls that are monitoring the accuracy of every single batch that we run in the laboratory. So positive and negative controls.
I can also add to that question on the sample collection device. So in our original FDA clearances that got us to the FDA general clearance, we were required to do a lot of robust reproducibility testing across lots of our collection device, across different laboratory sites. As we had more people integrating it into their assays, we started generating really robust data that showed on different types of assay platforms that you were able to get reproducible results. Like Shelley said, comparing back to Sanger sequencing, in some cases having to compare to a sample type that was already being used, like blood, where they wanted to switch to saliva. So we have a lot of really robust reproducibility data that we were required to generate to get to the general clearance that we have today. And I think that really just speaks to how robust and how much you can trust the samples coming out of the collection device, which is really important as you're integrating them into your pre-analytical workflow.
All right, fantastic. Thank you. We have some more questions coming in. Let's jump around a little bit. This one says, are there any temperature, humidity concerns, shelf life limitations, or any class 2 concerns?
So I can take that one from the collection device side. So maybe Shelley, you can speak to it on your test and how you have it labeled, but for our collection device on the pre-collection shelf life, so we've done very robust studies to support the labeled shelf life and the temperatures that it can be within, really aiming for that ambient temperature, being able to be stored between 15 to 30 degrees, and for the duration of the shelf life. So we had to do that initially for our submissions, and then we also have a really robust stability program where we monitor new lots being manufactured to ensure that the quality is still there for the devices that are coming off of our lines as we're manufacturing year over year. So it's something that we put a lot of effort into to ensuring that the shelf life is sound and maintained over time.
Well, and from our perspective, we rely on this data from OraSure and DNA Genotek. And from the user point of view, since these samples sometimes are collected and not returned always in a timely manner, we're very, very appreciative of knowing what the outside limits are and the expiration dates. Everything's clearly labeled on the collection device. So we're very cognizant of that. And we have less than 1% sample failure from collection at home going through the laboratory process. We get robust, high quality DNA.
Yeah, and I think that's really so that piece is super important too, Shelley. And on the post-collection stability side, we've also had to do a lot of robust testing to show how long the DNA is stable for through those expected transport conditions. So similar to the usability studies that I've walked you through, on our end, we needed to do analytical studies to really support that labeling of the number of days that it's stable at room temperature or through ambient transport. And like Shelley said, if you're integrating our device, you have the ability to leverage some of that data that we've already worked to generate to support in your test system, which is a value add of not having to repeat everything.
All right, wonderful. Thank you. We have time for a few more questions. This one says, how does the gluten ID report communicate genetic risk in a way that is understandable and actionable for consumers and healthcare providers?
So we have, through the FDA process, made sure that it is both understandable and actionable. Our reports are written in plain language, which is something that the FDA is very adamant about for a genetic health risk test that's going to be read and interpreted by the user at home. And so we report also graphically, and we have what we call our gluten ID risk meter that you saw earlier, and the gluten ID risk stratification chart that helps people not just read what the results mean, but see them. The results are actionable and just inherently because of the high negative predictive value of the gluten ID test. If the user doesn't have any of the genetic combinations that are positive for celiac risk, they can virtually rule out celiac disease with close to 100% certainty. And if they do have the genes, then within their level of risk, they can make actionable decisions if they're symptomatic about whether they want to go ahead and proceed down the path of celiac testing with their gastroenterologist or family practice healthcare provider supervising that. So we do have feedback from consumers already about how helpful it is to go into one of those meetings with the healthcare provider, knowing their gluten ID already, because genetic testing for celiac disease is not always performed, as you saw from the case study we presented earlier. And so they can go into a healthcare meeting with an actionable understanding of what their next testing steps should be. So that's the gluten ID test report, all supported by the collection device. We would be nowhere without it.
Great, thank you. I think we have time for maybe a couple more questions. So let's take this one. How does DNA Genotek support partners in integrating their collection devices into genetic testing workflows, especially for DTC applications?
I can start off answering this one, and anybody can jump in. But at DNA Genotek, we really take a consultative and individualized approach, similar to what I mentioned during the slide around the customizations and single order fulfillment. We're really looking to support our partners, understanding that everybody has unique needs depending on what the product is, the regulatory path, and the marketing strategy. Our support can range from something as straightforward as providing a write a reference letter to some of our previous FDA submissions so that the FDA can go back and look at the data so that you can leverage that rather than needing to repeat that work. It could also span to a really deep collaboration with an assay manufacturer. In these more involved cases, we really aim to work hand in hand to support the strategy behind the research and development, regulatory affairs, and even co-marketing strategies, depending on the case. For partners that may be launching a new product or really pursuing a new innovation that require that additional regulatory or technical strategy and maybe co-efforts in generating data, we often take that approach, and we're there with our partners every step of the way, whether it's assisting in some of the assay validation, generating usability data, supporting some of those analytical studies, as I mentioned. We really try to support and aiming really for the goal to be more than just a vendor. So we like to be a strategic partner that really can help enable the success from taking a test from bench to market. We're really passionate about what our products can enable. Like Shelley went through the importance of the gluten ID test, and it is really exciting to be able to work hand in hand as a partner with people offering tests like this.
Yeah, and just to add to what Tara said, we understand that it's really complex to go through the whole FDA process, and just based on our experience, getting our multiple collection devices cleared, we understand exactly what FDA is looking for in the studies that they would like to see in order to demonstrate performance, and just also understand the framework that FDA works is working in. So, for example, like just understanding how to communicate with FDA through pre-subs. So, because of our experience, and we have a dedicated team, on the R&D side with Tara and her team, and on the regulatory side with myself and my team, we're always there to support any of our customers that are looking to get a test, cleared with FDA, kind of supporting them and understanding what FDA is looking for from a regulatory standpoint, and, guiding them into, understanding how to, ask the questions to FDA, and also what are some of the, expectations that FDA has, because I think, at the end of the day, it's important to understand what FDA is looking for in order to get your, test cleared with them.
And I can certainly confirm that it is not just a vendor relationship. That DNA Genotek has been fantastic to work with since we ordered the first swabs to make our transition over to the new collection device. So it's been great.
All right, wonderful. Thank you. I think we have time for one more question. What are the strategic advantages of using U.S. FDA-cleared collection devices for companies developing new genetic tests, regardless of the status of the final rule on LDTs?
Yep. So I can take on this question, and if anyone has any additional thing to add, they can chime in. So the main thing is really that the performance has been demonstrated, and also that the product is safe and effective. So everything that all the studies that we run has been provided to FDA. So they've reviewed that information and have determined that the product is safe and effective, kind of a stamp of approval from them. So that's really what you're getting when you're using an FDA cleared device. Now, I'm not saying that, if you don't use an FDA cleared device, you're not getting a safe and effective product. It's just that, that's information that you may have to actually get from, the partners that you're working with, where, if you're using a cleared device, you kind of know already that you're getting some kind of level of certainty that the product is safe and effective. And then, for example, if you're using like our product, the OraCollect or Origene, then you also know that, it has also been demonstrated for at-home self-collection. So that's something that we've seen that FDA, has been really taking, initiative on to, kind of not only clear devices for like collection devices for use in a test system, but also specifically intended uses for at-home collection, especially after COVID. They know that, there are certain things that, and studies that need to be demonstrated for performance that you only see at home, where you might not see in clinic. So using a device like ours, where, we've demonstrated that at-home piece, that you're getting safe and effective, samples that are being collected at home. And the last thing is just an easy integration with, your test. So one thing we've seen that, FDA, especially with DTC tests, they're really looking at, from beginning to end, all the data needed to demonstrate that the test works. So when you're using an FDA clear device, you don't have to worry about the pre-analytical side. That's something that can be challenging if, having to, partner with a customer, relying on them and asking them to, provide those studies. And, that's something that they may not be willing to do, where, if the device has been cleared already, you don't have to worry about any of that, running those studies. So, and the last thing I would say is also that, as if you decide to just start off as an LDT, you may not need to use a, FDA clear device. But as, you're looking to potentially scale up to, sell your device, maybe as a DTC, where you then need to have an FDA clearance, it's always good to start off with a device that has already been cleared. Because, for example, if you start off with a device that hasn't been cleared, you may have to actually run, all your revalidation. But if you do use a device that has been cleared already, then, you're really just adding components to demonstrate that your test can be used as a DTC, instead of having to revalidate everything from beginning to end.
Yeah, and I think I can add, from being in the lab and from a scientist perspective, what I put value on, whether you're doing an LDT or taking a test system through FDA, is that if the sample collection device, such as OraCollect or Oragene, has been through multiple FDA clearance to gain that general clearance, status, it really tells me as a scientist that there's a lot of data to support this product. There's a lot of evidence, maybe, reaching out to us, and we can help you build confidence in that data, really understand what we've done and how it can support you as you develop your, say, LDT, in just reducing the risk of those failed samples or having challenges in the lab processing workflows. So I think regardless of if you're taking your assay through the LDT approach or through FDA, it really speaks to the data that's been generated behind the device, and how that can reduce the risks of sample fallout or sample rejection, somebody not knowing how to collect the device, all of those pieces that are really important for your upstream pre-analytical workflow. So that's something that I personally really put a lot of value in from a data side.
And I can add one more perspective to both Tara and John. As I mentioned earlier, we have a physician-ordered, pathologist-reviewed version of our test called Celiac DX, and the gastroenterologists that I've been talking with about the test, who've been starting to use it for their patients, like to order the test as they do traditionally for a laboratory-developed test, but they like their patients to collect the sample at home, and then they get the genetic results before. So with the general clearance for not just DTC, but also for prescription use, they have a lot of confidence in the collection device that would allow them to have the sample collected at home instead of bringing the patient into the clinic. So there's a lot that went into that device to make it very accessible, not just for at home, but also for clinic use, and a hybrid of the two when you have a physician order it and ask the patient to collect it at home. So I think everything that Tara and John said about all the work and regulatory efforts that went into making this the best device have really made it the best device. So.
All right, perfect. Thank you. We have one more question sneaking under the wire, so you can either answer it now, or perhaps you'd like to get back to this person offline. Yeah. No, they're asking if the cost of the device includes receiving the analytical reports.
Yeah, I saw that question. It's a great question. It's not something that we provide, an outline of all the studies that we've done to support a lot of those may have been done in collaboration with partners, right? Proprietary information. That's where, if you're going through the FDA path, the right of reference approach works really well because FDA can access the data and reduce the burden of studies that you need to do. However, if you are going through an LDT path, it's something that reach out to our account managers, and we have a lot of different white papers, app notes, collateral that we can share with you that are public, and that can help you when you're evaluating different sample collection devices to integrate into your system, or if you have any specific questions, we're happy to support. We have a really great product application support team that you can submit questions to around the compatibility. If you're developing an assay that is a newer technology and we don't have any collateral, there's a good chance that we've internally evaluated it, and we can give you at least some detail in terms of the risk of integrating it with that type of technology. So kind of a long answer, but yeah, reach out to us if you have specific questions, and we would be happy to provide you with any information that we have.
All right, great. Thanks, Tara. And we do have your contact info if you've left a question or comment, so we can have the team reach out to you. They've also kindly provided their contact information on the screen in front of you if you think of a question later or would like to reach out for more information. So I encourage you to observe that information before we close out in a moment. But this does bring us to the end of this webinar, and I'd just like to remind everyone that this webinar will be available on demand shortly following this presentation. Please watch your email for a message from today's clinical lab once this video becomes available. On behalf of today's clinical lab, I'd like to thank Tara Crawford-Parks, PhD, Jonathan Chan, BSc, and Shelley Gunn, MD, PhD, for all the hard work they put into this presentation. And I would like to thank all of you for taking time out of your busy schedules to join us. A special thank you to our sponsor, DNA Genotek. Their support allows today's clinical lab to offer these webinars free of charge to our audience. For more expert insights into the latest tools and technologies for the clinical laboratory, please visit our website at clinicallab.com. We hope you can join us again. Thank you and have a great day.